We're going to talk about viruses today. Before I explain the board, let me tell you what a virus is first. A virus is a small infection agent that can only reproduce inside the living cell of other organisms. Unlike bacteria or microorganisms, viruses are not considered fully alive because they cannot carry out basic life procedures such as metabolism or growth on their own. Instead, they depend entirely on the host so the cells can multiply. Okay, now I'm going to explain the structure of the cell. So let's start with the spikes over here. These spikes are protein structures embedded in the capsid or envelope. They can play a key role in attaching to and entering the whole cell. And now, capsid is a protective shell surrounding the genetic material. It is made of repeating protein substance called capsmeres. Capsid helps protect the viral genome and acts to attach to the whole cell. And then over there on the other side, we have another virus, but this one over here doesn't have an envelope. Unlike this one over there, has an envelope. The envelope are in some viruses and take from the host cell memory. The envelope can contain virtual proteins spikes that can help the viruses organize and entering the whole cell. For example, like HIV has envelope viruses. Oh, what else? Do I have to talk about this? Okay, so now we're gonna talk about the genetic material, which is viruses containing DNA or RNA. The genetic material carries the instructions for the making of the new viruses. One thing, genetic material. Viruses contain either DNA or RNA, but they can never have both. The genetic material carries the instructions for making the new viruses. Okay.
Hello, everyone. Next, we'll be talking about the different types of viruses. So there's two different types, DNA and RNA. And for DNA viruses, they replicate into the host cell's nucleus, while RNA viruses will typically replicate into the cytoplasm. RNA viruses can also mutate faster than the DNA viruses. DNA viruses need to use host cell equipment to transcribe DNA into mRNA for protein synthesis, while RNA viruses can just turn RNA into proteins. And then here's a little chart showing more of the differences between the two. Next, there's also the enveloped viruses versus non-enveloped viruses. Enveloped viruses have lipid bilayer membranes that surround the protein capsids, while non-enveloped viruses lack a lipid envelope. Outermost layer is the protein capsid. And then there are also some more differences between the two down there. And next, we'll also be touching on the impact on human health from certain viruses. And one virus that's been very prevalent over the past few years is COVID-19. Its symptoms vary depending on several factors, but it can be as small as a regular cold or as serious as developing pneumonia from it, which can threaten lives. Ebola is another example of a disease caused by a virus, with symptoms like the flu, in addition to unexplained bleeding, diarrhea, and vomiting. Measles is a third example of a disease caused by a virus and has symptoms like a cold as well, but rashes will appear on the body. COVID-19 is transported airborne or on contact with somebody else infected. Ebola is spread through the direct contact with blood, bodily fluids, or organs of the infected person, and measles can be spread through the air or on contact as well.
A virus and what structures it contains. So moving on from that now, we're going to talk about the virus life cycle. Number one is absorption. Absorption begins when a virus invades a cell of a host that is susceptible. Some examples of susceptible hosts can be elderly individuals, infants and children, pregnant women, those suffering with HIV or undergoing chemotherapy, and people with chronic health illnesses. When a virus absorbs itself into a host cell, Its spikes will attach themselves to specific receptors in the cell's membrane, like shown in this picture. Here are the receptors, and here are the envelope spikes. And as you can see, the envelope spikes are attaching itself into certain receptors in the host cell membrane. Number 2 is entry through endocytosis. This happens when the virus is already inside the cell and is surrounded by a vesicle. And then number 3 follows, right here, where the virus's structures like the envelope, spikes, and capsid are dissolved by enzymes in the vesicle to free either the DNA into the nucleus, as seen here, or RNA into the cytoplasm. Number 4 is synthesis. Because the nucleic acid, either DNA or RNA, was injected, the viral nucleic acids now have control over the host's metabolic functioning. This means that the host cell is manipulated into synthesizing protein, RNA molecules, capsomeres, and spikes for more viruses, to then number 5, assemble. In this example, the spike proteins, aka glycoproteins, are inserted into the cell membrane in order to form a viral envelope. The capsid is then formed by the new RNA and the new capsomeres. If the original virus had DNA material, then it would stay in the nucleus. Therefore, it's called a provirus because it integrates, as you can see, into the host cell genetic material. Then number 6, the virus gets released. Envelope viruses like this picture bud off of the membrane. This is called exocytosis. As you can see, a small little pouch is formed, and then it pinches off, and then the pouch releases the virus with its envelope without destroying the cell. If it was a naked virus, then it would destroy the cell because naked viruses are more virulent than enveloped viruses.
Hi, so now we'll be talking about vaccines and how they are our defense against viruses. So first, I'll start off by saying what a vaccine exactly is and how it works. So a vaccine is a weakened or inactive form of a pathogen that is injected into your body to allow your immune system to produce antibodies to recognize and fight it off without causing illness. Which now brings us to the reason of why vaccines are important when treating viruses. If the person is exposed to it in the future, the cells will be able to recognize it quickly and produce the antibodies to fighting it off, preventing it from getting severe. So then here is a timeline of the history of vaccines, but we want to pay particular attention to the one that occurred in 1796, which is the vaccination of smallpox. The vaccination was created by Dr. Jenner, and he was able to collect the matter of a cowpox sore and inoculate it into an 8-year-old boy who later then became the first person to get vaccinated against smallpox. So now on to the different types of vaccines that there are. So we have live cells or viruses vaccines, which contain a weakened yet still living form of the virus, and because they are so similar to the natural infection, they are able to produce a strong and long-lasting immune response, and it's usually just 1 or 2 doses and you'll be all set. Then there's the inactivated vaccines, and these use a killed version of the germ to stimulate the immune system. And since the germ is not alive, the immune response may be less strong, which means they'll typically need more than 1 dose. And an example of this would be the flu vaccine, which typically is needed every year.